Now, how is metamorphosis controlled by the hormones in amphibians? One model that was proposed very early during the studying of metamorphosis in this animal is called the threshold model. The threshold model would indicate or would suggest that the different events on the body of the animal are triggered by different concentration of thyroid hormone. Now, this model can explain some events, but not all the events. And in fact, the reality is a bit more complicated than the very simple threshold model would suggest. Let's see how the model would apply. Well, first of all, we know that some organs need to be developed very early, and they require, in fact, they require for metamorphosis very low concentration of thyroid hormone. This is the case of limb development. So the tissue that need to outgrow the limb require very low concentration of thyroid hormone to produce the limb. This is if you think about it, it's logical. The limb development during metamorphosis is a very early event, and this is because you want to have a way of locomotion. Imagine if the tadpole would lose the tail before producing the limb. If the tadpole loses the tail before producing the limb, it will not have any means of locomotion. It will become like a blob waiting in the water to be eaten. Other events, like for example the resorption of the tail or remodeling of the intestine, happen later during the metamorphosis, and they require higher concentration of the thyroid hormones. So this model would predict these events. However, metamorphic changes do not depend simply by the concentration of the blood concentration of the thyroid hormones. In fact, the thyroid express and produce and release in the bloodstream thyroxine, T4. And here on the right, you have the formula of thyroxine, which I will not ask you to remember. However, the concentration, if you want, of the thyroid hormone do not simply depend on the production of thyroxine from the thyroid. In fact, the active form of the thyroid hormone is free iodothyronine, or T3. And T3 is converted by specific enzymes that are called type 2 deiodinase in the target tissue. So every target tissue will express different quantity of this enzyme, which will convert T4 to T3. Just if you are passionate about biochemistry, you will notice that the difference between T4 and T3 is the absence of one iodine group in this aromatic chain, or aromatic chain. Moreover, the concentration of the active form of the thyroid hormone, T3, in a tissue, in an organ, depend also on the organ expression of the degradation enzyme, the type 3 deiodinase, that convert T3 into T2. The T2, which has only two iodine group, is inactive. So now you see that things are more complex than what the initial simple threshold model would predict, because the concentration of the active form of the hormone T3 depends from the production of T4 from the thyroid, but also depends on the rate of conversion from T4 to T3 in the target tissue, and as well as the rate of conversion from T3 to T2, which is inactive, in the target tissue. So, moreover, to add complexity, T3 need to bind specific hormone receptor, thyroid hormone receptor, which in this thyroid hormone receptor, they have higher affinity for T3 than T4. That's why T3 is actually the active form, because this receptor, they need to be need to bind T3 to become active and therefore induce expression of target genes as an affinity, a higher affinity for T3 than T4. So metamorphic events therefore are triggered by the not only by the production of T4, but the rate of conversion of T4 to T3 and the rate of conversion from T3 to T2 in the target tissue, as well this expression of the thyroid hormone receptor in the target tissue. So how much T3 do you have in the target tissue? How much TRs are expressed in the target tissue? So all these elements contribute together or play a role together to determine whether a tissue responds to a particular concentration of T4 in the blood, and whether a tissue is responding early or later during metamorphosis. But of course, this is not, there is another layer of complication that we need to consider, and we need to consider the fact that, for example, the thyroid receptor, they're not only one single type, we have multiple types, and in particular we have alpha and beta. Thyroid receptor alpha are expressed in almost every tissue, almost every time, even before the thyroid is formed. However, thyroid receptor beta are expressed only when thyroid hormones are produced and they reach the target tissue. And they form, in fact, they form a positive feedback loop. The more thyroid hormone reach a tissue, the more TR beta are expressed. And both TR alpha and beta form active dimers with retinoid receptor. So the TRs and this retinoid receptor are XR, 4 dimers. You can see them here. So form the complex, and this complex sit on target genes. And in fact, the receptor with the retinoid the thyroid receptor with the retinoid receptor together, they actually block the expression of target tissue. They block the expression of target tissue until T3 arrived. When T3 binds this complex, it activates the expression of the target genes. So the thyroid receptor and the retinoid receptor already sit on genes that should be activated. But when they are there alone, they repress those genes. But when T3 arrived, it formed a complex with the TR and RXR so that the genes can be activated. Now, one may ask, how come some tissue respond to thyroid hormone by producing, for example, new tissues like new neurons, new muscles, and so on, while other tissue are eliminated, like the tail? Well, nobody is totally clear about how different tissues respond to the same signal, thyroid hormones. But what we know is that for sure, the specific kind of response that a tissue has is already encoded in that particular tissue. How do we know that? Well, we know that because we can do some transplant experiment and see what's happened to the tissue that we transplant. So, for example, in this particular experiment here on the left, a tail has been transplanted in the region where new limbs are supposed to be formed. Now, if the information about the degradation of the tail is already encoded in the tail, this particular extra tail should be removed during metamorphosis. Otherwise, if the information about producing new tissue is encoded in somewhere else and not in the particular tissue that we are talking about, then the tail should grow more, like a new limb is growing during metamorphosis. But what's happened, in fact, is that this tail, which is out of place, received thyroid hormone information and it started to be reabsorb like a normal tail. This suggests us that the information about the particular output of the metamorphosis when the tissue receives the hormonal information is already encoded in that tissue. Similarly, in this other experiment on the right, what was done is to transplant on the tail an eye from a donor. Now, the eye is supposed to form new lid during metamorphosis. However, because it's on the tail, maybe it gets reabsorbed. However, what's happened during metamorphosis to this animal is that while the tail is indeed reabsorbed, the tissue containing the eye is not reabsorbed, but undergo metamorphosis and indeed develop a new lid, suggesting again that the specific information about what to produce when exposed to the hormonal information is already encoded in the target tissue.